Okay, this is your video tutorial on how to do experiment number 3. I'm coming in. So what we'll be looking at in this lab is, or in this experiment, is the difference between a pelvis that is more biased to the left side and a pelvis that is more biased to the right side. So if you recall, the pelvis is your hip bones. In any other normal animal, that should not be asymmetrical. So for us, it's symmetrical. That means it's the same on both sides because your two legs are also the same. So as the sticklebacks begin losing their pelvis, it kind of goes through like a middle stage, which we've been calling the reduced, where it's no longer full, but it's also not completely absent either. And at that time, it'll be kind of more to the left or more to the right. And then when we look at that, we can make some assumptions about how the genes that are creating that that pelvis, how they're kind of being altered. Remember, if you watch the background video, you learned about the PIDX1 gene. So I'm going to skip the staining and quickly to scoring the fish. Or if you check out this picture here, this is very important for how to do it. You see that if the top part of the pelvis is bigger, then it's called a left bias. And if the bottom part of the pelvis is bigger, then it's a right bias. So here we go to look at some actual fish. Woo, strain it, pour out the alcohol, and let's get under the microscope. Okay, ventral view. So, sorry, my magnifier is weird. We said if the top is bigger, it's left bias or right bias. Let's check. Okay, left bias if it's bigger on the top. I do believe this guy's a little bigger on the top, so left bias. Okay, get rid of him. Bring another. Again, I'm seeing bigger on the top, left bias. Third guy. Definitely bigger on the top. There's nothing on the bottom there. Left bias. Okay. I'm still feeling the left bias. This time, oops. I'm possibly feeling a right bias this time. It looks a little bigger on the bottom to me. Oh gosh. Left. Anywho, you'll continue this all the way through for Bear Paw Lake and for Frog Lake. When you get done with both of your scoring, then you will verify your results. You'll click on to view the graph, and then after you see the graph, you'll be able to go into the quiz questions. And that